So today we'll be talking about the basics of spectral analysis for sleep research. And I know many people have seen spectral analysis lots of places, but might not really know what it is or what's behind it. And the goal of this is not to teach you all the math to make you understand everything, but to have you come away with an appreciation of what it is, what it's good for, and some things to maybe think critically about it, and also how it might be used in sleep research. And so all of this stuff is related to a paper that we did in physiology. You'll see the link on the side, and a few other things that I can point out, some resources for you at the end. So let's get started. So how can we go about describing complicated things? You see something like this Jackson Pollock painting, and you can imagine a thought experiment where I said you have someone in a different room who does not see the same painting. How would you get them to replicate this painting? How would you describe it? And that's not a very simple problem with a simple answer. And so the question of that is, well, we could sort of first try describing it, right? You could describe little bits of this and verbally explain what's going on. Or, we're living in a digital age, we can sort of take the computer approach and break everything down into little discretized pixels. And then you have millions of these things if you want to do it super high resolution, and you describe what the color values are for each of the coordinate pixels. And there are pros and cons to both of these methods, but it's still not a very simple question. These are different ways of approaching them. We have the same problem when it comes to looking at the EEG. We have this series of squiggles. How do we describe them? That's not a really easy question to answer either. It's same level of complexity, things changing all the time. How do we go about this? Well, we can ask, well, how have people gone about this in the past? And we can go back to the first experiments from Hans Berger in the 1920s, where he was first looking at these squiggles and trying to sort of make sense of them and how they changed as a function of things that were happening, in particular, someone closing their eyes or going to sleep. And one of the first approaches that he actually did use is you can go through each little up and down little blip here and wave in this and categorize the the size of the wave and how far apart they are. And that's a very precise way, and you could probably reconstruct things very well, but that's actually quite difficult. So what's another approach to describing really complicated things? And one answer to that is the idea of a basis function. Now, we can go back to art. Anyone who's learned how to draw knows that one of the things you can learn is to say, if I have a complicated shape, I can break it down into simpler shapes or simpler building blocks. And so we call these basis functions in mathematics. And in the case of 3D art, which you can see on the right, it's built in. They're called primitives. You can have a cube or a sphere or a cone or a pyramid, and you put them together and add them and subtract them in multiple ways, and you can use those to build very complicated objects. And that's essentially what's done in 3D graphics, among some other things. So really, a basis function is just a simple building block to make something. And they're a mathematical object used to form something much more complex. And we can keep it at that level. And the cool thing about that is we can decompose these complicated objects into the basics components to sort of get a useful summary of what the structure they are of that complicated object is. So we see Legos here will be our example moving forward. So let's say we have a very complicated object and a very scary and carnivorous one as well. And how can we represent this as a function of simple things? Well, first we have to choose our basis object or basis function, which in this case, our simple building block are Lego. And here is our representation. We can convert what we observe to Legos. Now, right now, we have a limited number of blocks that we're using, but you can imagine the more blocks we use, the better and better representation we can have of the object that we're trying to describe. So that's sort of an important concept in basis functions. You want something that if you use enough of them, you can do a really good representation of the thing that you want to describe. And even better, as the number of blocks approaches to infinity here, you could imagine getting to the point where you could describe every single little part of this complicated bigger thing. Now, what's nice about when you decompose things into basis functions or building blocks is that you can take now, instead of describing, saying, here's a Tyrannosaurus Rex, you can break it up into all of the different parts that compose it. And then you can take them and tally them up and summarize them to say, well, I have a certain number of these, 2x2 green squares and 2x4 flat pieces. And it's a really nice summarization that you can have of the complicated thing that you're trying to do. So what's interesting here too is, and something that we should note, one of the complicated things about this is that the summary might not be unique, and we'll talk about that a little bit later. Here's a Lego 3-in-1 set where you have the same blocks that can build 3 different things. So it's important to note that these sorts of summaries talk about the composition of things, but maybe not the exact order or the way things are going if you look at them in certain forms. So let's get back to our good friend Hans Berger in the 1920s. Here he is with his lovely young son. And if you look at the history of the electroencephalogram, and in particular sleep, people like to experiment on their children, which is just an interesting trend. But one of the things you notice is that on the bottom, you have the actual trace. And here is what you see here is the actual EEG trace. And he compared it to a 10 hertz pure sinusoid. And he noticed that when eyes were closed or as people were starting to fall asleep, you had this really clear sinusoidal type rhythm, and it was the first one that he discovered, so he called it the alpha rhythm. And that's how we sort of got the basis of the EEG bands that we use today. And moving forward, we say, you could say, well, maybe these sinusoids or these oscillations can be a good basis function for decomposing the spectrogram. And in fact, in EEG literature, that's exactly what we do. We break up the complicated signal into different components at different frequencies and give them names. And that's a very easy way of saying, here's this very complicated shape that can be broken up and represented in a simpler format. Now, wouldn't it be nice if there was a whole section in theory of mathematics devoted to doing such a thing, breaking things into sinusoidal parts? And in fact, there is. It's called Fourier analysis, and it was done by Jean-Baptiste Joseph Fourier in the 1700s and 1800s. And in the process of trying to understand how heat propagates through a metal sheet, he actually came up with this concept of Fourier analysis. And the one thing that you really, if someone say Fourier transform or Fourier analysis, is the thing that you want to remember is the idea behind it is that any infinite length repeating signal can be represented by combining sinusoid. That's a simplification, but that's really all you really need to know behind it. So if I have a signal that repeats itself over and over and over and over and over, I can do a really, really, really good job of representing it using sines. So in this way, how does Fourier analysis apply to our problem? Well, in our case, going back to our Lego example, instead of the T-Rex that we're trying to represent, we have this EEG signal. Instead of Legos, we have a whole group of sinusoids that we're adding together. And then instead of our list that tells us the number of block as a function of type of block, we have something called the power spectrum, which tells us how much each of these sinusoids really contributes and in what way it contributes as a function of the frequency. So a higher frequency means the sinusoid's going up and down faster. So what's really nice is you can put it all together and you can see that this works in theory, that you can have a shape like this square wave on the top and show how a bunch of sinusoids put together, those 3 sinusoids, start approximating this really nice, very non-sinusoidal type repeating signal. So ultimately in this talk, we're going to go from the fact that we're looking at EEG in the time domain, which is the waveforms, and we're going to move to the idea that we can view an entire night of sleep as a really nice thing called a spectrogram, which is the power spectrum over time. So overall, we're going to be talking about spectral estimation, and that's sort of saying, if I have my basis functions about frequency, how do I actually compute them and what are the implications? So we can think about, we spoke about building these things up, but we can talk about taking the signal and breaking it apart now. And we can say spectral estimation is breaking down the signal into its frequency components and asking sort of how strong should the sine waves at each frequency be and where should they be located and so forth. And we're talking about, again, the terms, the time domain, which is the waveform over time, and the frequency domain is the power spectrum, or how it looks when we are converting this thing into frequency. And you can think of this akin to just how a prism can take light and separate it into the different waveforms, or if you have one of those old school graphic equalizers, you can break up sound into the different low, medium, and high bands. So what we now are going to start talking about is waveforms. And there's a couple of terms we want to use. Periodic just means repeating, and stationary means not changing. In this case, not changing its properties. So if we have a repeating time series of repeating signal, you can see how, while that looks complicated, it is actually just the sum of these different time events, these different frequency events, 1 hertz, 10 hertz, and 12 hertz, right? And so what's nice about that is we can convert that into what we call a power spectrum, which on the x-axis shows the frequency and power is on the y. And since we have 3 perfect sinusoids making this, we have 3 vertical lines at the frequencies at which the sinusoid occurs. Now, you might say, well, things like the EEG change over time, and that's correct. And the assumption when you're taking a power spectrum is that the contents within it have the same frequency content, and they're stationary within what you're looking at. And in Fourier analysis, we assume that everything is infinite, but we're not actually looking at infinite data, we're looking at finite data. So how do we tackle something that changes over time? Well, pretty simply, we break it up into bins, and we assume that each of the bins are stationary. Then we take the spectrum for each of those bins, and we get something like this, which we call a spectrogram. And this is called the time frequency domain when we're looking at how things change over time and over frequency. The way you read this is the hotter colors have stronger power, the cooler colors have weaker power, and time is on the x-axis and power is on the y-axis. So just some terminology that I want to go over, because I think this is often confused by a lot of people, and I'll just do this briefly. But from a theoretical standpoint, we're talking about the math that goes behind it. There's the Fourier transform, which is sort of the math behind all of this, that assumes sort of infinite periodic data for the most part. And it's really good for building theoretical models. Now, in the real world, we don't have infinite data, and we have to look at little chunks of data. So we use what's called a discrete Fourier transform, and that's really good for real world data analysis and good for when you break things up into the window. Now, one term I think that gets confused a lot is the FFT, and it's important to know that the fast Fourier transform, or the FFT, it's simply a specific computer algorithm that implements the discrete Fourier transform, and it's really good for computers. So when you're talking about the FFT, you're talking about a computer algorithm. When you're talking about the thing that it produces, that's the output of a discrete Fourier transform. And in terms of the output, we have the spectrum, which is the power at each frequency. The power spectral density is just a spectrum that's normalized, and the spectrogram is just the spectrum changing over time. So I think this is really important to know, because I certainly see these terms confused in many cases. So why is this good for sleep? Well, now we imagine you have this data, the signal over here, which one might say, oh, well, this is an EEG channel that's corrupted by ECG. Well, if you break it up, it's made out of different components. There's the actual EEG component, there's the ECG component, and then there's the line noise at 60 hertz. And so what's nice is that if we view this as a spectrogram, we can see that each of the different components that we care about live at different frequencies, right? So you can see that, I'll use my pointer here if we can see this, right? That the ECG lives way down at the bottom at this lower frequency, and then the EEG this is an alpha signal occurring right around 10 hertz, and then line noise is over here. So this is a nice feature of these things where you might care, depending on the analysis that you're doing, you might feel you have to come up with an algorithm to remove the heartbeats from the EEG. But in this case, if you're looking at spectral analysis, and again, depending on what actually thing you're looking at, it all separates out. So that's a nice benefit here. So typically, why hasn't time frequency analysis taken off in sleep medicine? Well, essentially what's happened is we like to look at things as a function of power in these discrete bands, and that makes sense. But that sort of blurs what's going on. And I know even in the older versions of some of these machines or systems, you used to be able to get a nice spectrogram that came out of that. But it was typically they used a periodogram, which we'll talk about, or a single taper spectrum, which is not very good because there's a lot of noise in that. So it was kind of a feature that was left on the wayside. And what we'll be working towards is something called the multitaper spectrogram, which I sort of feel is like taking this and putting glasses on, which is a way of just really bringing out and reducing some of the noise and some of the properties that make it difficult to resolve things. So now we're going to talk a little bit about how these things are computed, the actual spectral estimation, and why things can be easier or harder to show us what's going on in the data. So the basic analysis that you do, if you take, usually the output of the FFT gives you something called a periodogram, and it's actually a poor spectral estimator. And why is this, right? Well, as we said, we assume that there's infinite data when you're doing this Fourier transform, but we're doing a discrete Fourier transform, which means we don't have real data, infinite data. We have a chunk of data. And when you have a chunk of the data, it turns out it's and if we had infinite data and we computed the actual spectrum of it, we'd have this beautiful perfect line because we have a 10 hertz sinusoid here. But in reality, we have finite data. And if you look at that, it actually gives us this complicated thing that does not look like a vertical line at all. And what you'll see, and we'll come back to this, is that when you compute the spectrum of real data, you have a big bump in the middle called the main lobe, and that's the main frequency that you observe. And then you have this ring out off to the side, and those are called side lobes. So those are just terms. So rather, what we would expect theoretically is something like this, but in actuality, we have something that looks like this. And why is that? Well, it turns out that when you're looking at finite data, it's the same as taking infinite data and multiplying it times this square window over here that's 1 during the period where you observe data and 0 everywhere else. And so it turns out that you're not just taking the Fourier transform of this thing, which is the sinusoid, which is a line. You're actually also kind of taking the transform of this square window. And if you recall our initial example that we showed trying to make that square wave, you need lots and lots and lots and lots and lots and lots and lots of sine waves to make that up. So when you smush that together for real data, you get something that looks like this. And we'll talk a little bit more about the main lobe and the side lobe. And ultimately, that's why you don't get something that looks very nice when you're looking at short periods of data. So in general, what this also means is that it turns out that this periodogram, that is what the standard output that people usually use when they say they do an FFT analysis, it's biased. And in mathematical terms, that means it's always going to differ from the ideal, the expected spectrum at each time. And it gets really bad when the data length is short. So one of the things that we're going to talk about is that there's sort of 2 types of bias, 2 types of badness that you can get here. There's this main lobe here, and there's the side lobes here. And the side lobes basically say, I'm going to take all of the power that should be in this vertical line and spread it out on both sides, on all sides of the frequency, and take this concentrated frequency that I expect to observe and push the power and blur it all out. And the same thing is this main lobe here is this big wide lobe that tells you where the frequency at which the most power is, but because it's not a line, it also blurs things that are close together next to it. We'll talk about that shortly. And so one of the reasons that we have this ringing here is because of this square wave shape. It has very sharp edges, and it turns out that if you take the Fourier transform of something with sharp edges, it requires lots and lots of sinusoids to look very, very good. But what if we smoothed out the edges? And so what you end up doing is you can use this thing called the taper function, where you're essentially making the data that you observe 0 at the beginning and the end. So we move from something that's just the data cut off to something that starts low at the beginning, smoothly gets bigger, and smoothly gets smaller. So we're just multiplying it times the smooth window. Well, it turns out that that smooths thing out and reduces some of this broadband bias, and look at what we have. So we have some changes here. We see that these side lobes have gone down, which is good, but it blurs the main lobe a little bit, and there's a trade-off on this. But usually, if you when people do the majority of things that you see use a tapered periodogram or a tapered spectrogram to reduce this, to make things a little bit better. And so the other thing that happens is that when we look at the output of the data, it's also very noisy. There's a lot of variance to it, right? And so it turns out that we want to reduce that. And I'm going to skip some of this we can come back and talk about it this question. But here's what happens. Here's your periodogram on noisy data. And then we take that taper of that here, and you can see that what happens is these side lobes go way down, but it's still really noisy. So how can we remove this variability? How can we reduce that? So we're going to work towards something called multi-taper spectral analysis, and it was developed in the 80s by David Thompson. And it basically reduces the side lobes and also reduces the variance that we have in our estimate of what's going on there. And so how does it do that? Well, imagine you're doing a regular experiment with noisy data. How do you get a good estimate out of that? Well, you repeat the experiment again and again, and you average the data across the trials, and then you get, hopefully, the noise cancels itself out, and you get the signal that you're looking for. The problem is we only have one, we're not necessarily looking at multiple trials. You could do this with the actual data, but really, wouldn't it be nice if there was a way to do this if we only had one trial? And so we're going to apply this concept of tapering and multiple trials to spectral data, and this will get us the multi-taper spectrogram. So what we're going to use, the shape of instead of just using one taper, we're going to use multiple tapers. And the shape of these things comes from something called the DPSS sequence. It really doesn't matter what this is, but at the very basics, you need to know that these are different shapes, different taper shapes that we multiply the data by, and we're going to average them together. But these are very special shapes for a couple of reasons. They're shapes that were designed to taper things so that you keep most of the power in the main lobe. And they also have a mathematical property called orthogonality, which means they produce projections of the data that are uncorrelated with each other. That means you can average them together. So in simple terms, these are really good functions for smoothing out the data so that it is able to keep all of the power around where the central frequency is. And they have a special property that they become uncorrelated with each other, so you can average them together. So really all multi-taper spectral analysis is we take a single segment of data, we multiply it by these special group of tapers, and then this is the data that we come up with. We compute the spectrum for each of those, and we average those spectra together, and that becomes the multi-taper spectrum. And so this is what we use. And so this is really the 3 steps that we have here. So why is this good? Well, you can see right here, there's the single taper spectrum, and then we do the multi-taper spectrum. We've removed a lot of the noise in here and can see things more clearly. And if you take slices of the actual spectra at each time, you can see that the multi-taper in red does a great job of reducing some of the noise that you're not able to get with the single taper spectrum. And so there are a bunch of parameters that you need to use when looking at the multi-taper spectrum. And there are 2 things that you really want to look at. First is the size of the data window that you're looking at. And then there's the frequency resolution. How wide do you want this main lobe to be? And there's some equations that you use to get these that we can talk about later if people are interested in. But essentially, you have to ask a couple of questions here. You have to ask, how big of a window can you have? How long of a window can you have where you believe the data are going to be stationary within that window? So if you're trying to examine something that's changing quickly over a short amount of time, you might want to have a small window. If you're looking at something like that takes place over a long period of time, you might be able to have a larger window. And then the next thing you want to look at is frequency resolution. And the idea is to say how long, how far apart are the different components of your signal apart from each other in frequency. So it's what's the smallest difference between the peaks that we want to be able to resolve. So if you have a frequency resolution of, and we'll talk about this explicitly, if you have a frequency resolution of 2 hertz, if you have 2 sinusoids that are within 2 hertz of each other, that big main lobe is going to blur them together and you won't be able to see them. So we have basically, this is sort of the framework that I suggest for selecting your parameters. So you set the size of your window by determining the length of time over which you assume the signal is going to appear stationary. And then you set the frequency resolution given the structure of the data and how big the bumps of the things you want to do are separated away. And so then you can compute these 2 things here, this TW and L, which are things you put into the Multitaper spectrogram code that we will be sharing with you. And again, we can get into a little bit more of what this means. But this allows you to change the properties of your spectrum so that you can really explicitly understand what's going on and how it relates to the data. So what you can see is when you change these properties, it changes the spectrum. And what this is doing is saying, if I assume that the space between the oscillations I'm looking for is further apart, I can have a larger spectral resolution, which blurs the data and makes it nice and smooth, but you might not observe some very small differences. So let's get to the main point of this, which how can we use this to better characterize the sleep EEG? And so just to sort of review, what we have here is your standard hypnogram, right? That shows the stages as a function of time. And then here is your spectrogram over here. And the way, again, we read this as time is the x-axis, frequency is the y-axis. And when you look at the frequency, this is what we observe when you observe power here. And if this is at 10 hertz, this is our alpha, and our delta is down here, theta and sigma where the spindles live. So what we're able to do is, even if you don't know how to read this thing, you can see that it repeats and mirrors the structure of the hypnogram above. And if we were to look at a whole 10 hours of sleep just using the waveform trace, you can certainly see some structure here with the amplitude corresponding to the slow wave aspects here, but there's really not a lot that you can see. So by looking at the full night multi-taper spectrogram, you can, within an entire eyeful, see everything that's happening in the sleep macro architecture. And so one of the things that's interesting is that you can see that there's some big differences. These are 2 different healthy young subjects. But you can see that those repeat reliably with respect to the macro architecture that you see in the traditionally scored hypnogram. And so let's take a look at how to sort of read this. What you have here is a section of good wake. So what we're looking at here is the spectrogram, and you don't see a lot of activity. And the second the subject closes their eyes, you see this strong alpha. You see this strong alpha oscillation over here, and then you also see this harmonic over here. And we can get a little bit into what harmonics are. That answers the question is, what if the basis functions that you use don't quite fit? So if I'm trying to fit this square and my basis function is a circle, well, what can I do? Well, I can start with a big square. I can add some little square, add a big circle, some little circles, and keep adding circles, and I'll get a reasonable fit. But if I look at this over time, what you'll see is that I'm going to get a bunch of fits from this little circle as opposed to one fit if my basis function was a square. And so this is the same thing when you look at these. I'll go back here to this slide on harmonics, that it turns out that the alpha oscillation isn't an exact sinusoid. So in trying to fit this, you get these sort of ghosting harmonics above. Doesn't mean that there's an actual signal here, but it just means that our sinusoid isn't doing as good of a job as we want fitting this function in a reasonable way. And there are ways to go about that. So now here is active REM. And it's important to know that when you move during sleep, it will make these motion artifacts here. And this is what motion artifacts look like. So you have little bursts of alpha here when the eyes are, you have the eyes open and they're closing and they're opening and closing their eyes here. So you see it going from alpha and back and motion artifacts. So this is what sort of an active wake looks like. Now let's take a look at what REM looks like and as a non-REM looks like. And so sort of definitionally, we expect non-REM to be an increasing slow wave activity. We also expect to see spindles. And so here's what you see. Here's a subject that goes from wake to stage 1 to stage 2 to stage 3. Here's their alpha, which goes away during stage 1. And then you start to see this sigma activity, which are the spindles. We can talk a little more about that. And that signals stage 2. And then at stage 3, those sort of go away and you have the strong increasing delta and some theta power there as well. And what's interesting about this, we can sort of call this shape the non-REM motif. We see this image here. And here's someone who has a little bit of a fragmented non-REM. And you can see this motif sort of 3 different ways. In this first period here, you see the standard descent into non-REM sleep. Then they have an arousal here. They wake up. You see there's a motion artifact and some alpha. And then now you see them descending here into non-REM again, because you have the rising slow waves, the spindles that come and go. And then you see a lightning. You see this almost with this 3 over here is the mirror image. They're lightning here. Their slow activity goes down and their spindles increase again. So we can sort of remember this picture, and this can allow us sort of in an eyeful to really characterize non-REM very quickly. We'll next speak a little bit about sleep spindles, which is a subject that we've in my group have been really interested in. And so now you see the sigma band here. And if we zoom in on it, look what we see. We see these little blobs here, and they correspond very well with scored spindles. And so you can even see that for subjects that have fast and slow spindles, you have blobs at different places. And it turns out that anytime you have a little transient, short little oscillation, it will cause a peak in the spectrogram. We call that a time frequency peak that will cause one of these blobs to occur. So let's talk a little bit of why spindle detection into the time domain is difficult. So what we have on the bottom is an observed simulated period of sleep. You have a slow component here, a noise component, and a spindle component. And as we watch this go, I'll move my mouse out of the way, you can see that depending on where the spindles are on this bottom and how strong the noise is, that either makes it easier for us to see it or harder for us to see them. And so if you can imagine going and visually scoring spindles, there's going to be a lot of luck, and it's going to bias us to only those large spindles that we can observe during periods of low noise. So why is this relevant? Well, if we do the same simulation in the time frequency domain, even if we throw a lot of noise into it, you can see that these blobs are very robust. These time frequency peaks are very robust and easier to observe. And so what we end up seeing consequently is that if we look for spindles, and this is where a technician scored sleep spindles on this trace, you can see that they align with these time frequency peak blobs over here, but we also see these other time frequency peaks that are not scored. So this is a subject of a paper that we just had accepted in Sleep and should be coming out soon, where we show that if you look for these time frequency peaks, instead of looking at traditional time domain spindles, not only do you have 3 times the number of events at least that you observe, you do a really good job of being able to detect them. And if you look at things like night-to-night variability, the rate for a subject that was recorded on 2 different nights, how does their spindle rate relate from night 1 to night 2? If we look at traditional spindle detection, we have a correlation of 0.6. But if we look at detecting these time frequency peaks, we have a Pearson's correlation of 0.98. And that's impressive considering that there's 3 to 4 times as many events per things that we observe here. So if they were noise, they would not be helping us. So let's move to REM, and we'll move along here because we're running out of time. So let's move to REM here. One of the interesting things that we observe during REM, which is not really noted for in any of the scoring literature, is as the subject goes to REM, we see this very low long period alpha bursting. And this has been noted in the scientific literature in specifically work by Prerau's group. But we call this peri-REM alpha bursting because it occurs before and after REM. These things can be pretty long, and they occur. And again, this relates to some of the work relating to tonic and phasic REM as well. But it's very, very clear on the spectrogram. And because it occurs over long periods of time, it's something that you would not be able to see easily in a time domain trace of a 30-second window, right? This is happening over 30 minutes in the middle, and it makes it very, very clear. So if we were giving this in person, I would have everyone shout out what they believe, but just to give you an example, we can look at a night of sleep and look at sort of the templates of the different stages here. And we can sort of go through and say, okay, what's this period? Well, this looks like some active wake followed by quiescent wake. Over here, we see 2 non-REM periods, one going forward, one going backwards. Here we see REM that has that alpha bursting there. And then we see REM, non-REM, REM, non-REM, REM, non-REM, REM, non-REM, a little bit of wake here, back to non-REM, little REM, non-REM, wake, and then we're done. And now you've just basically scored your entire sleep record, at least enough that to compute an AHI for in about 15 seconds. So we see this as highly useful as a way of at least clinically doing a quick estimation of things like total sleep time. But more importantly, you get a really good sense of all the electrophysiology of the subject that you're looking at. Now, finally, just to wrap this up, you don't have to just look at the EEG signal. You can look at the EMG signal. And you can look here, this is the chin EMG during wake, and you see this broadband activity, and then you compare it down to the bottom to the little twitches that you get during REM. So this is a very powerful tool. Moreover, you can also look at what happens during respiratory events like sleep apnea. And often you see these really interesting bursts going on here that I think are something that's a subject of future study here. So I'm going to wrap this up now, and thanking you all for your time. And what I would like to say is, if you have any, if you want to delve into this further, you can see on the links that we have a full tutorial on the Multitaper spectrogram on my site, which is sleepeeg.org. We have a spectral scoring manual that you can look at that sort of distills this. And very shortly, we'll be releasing some software that will allow you to go in and score these things manually.